Let me welcome all of you to the departmental seminar today for EEB. I'm very pleased to introduce Luke Weaver, who is a postdoctoral fellow who started here in September. And Luke is working with my research group that's interested in the influence of mountain building on mammalian evolution. Now, Luke got his PhD earlier this year during the summer.
Recording in progress.
From the University of Washington, where he was in the EEB department there and working with a vertebrate paleontologist, Gregory Wilson Mantilla. And he and Luke worked on some of what you'll hear about today. He worked on this unusual group of mammals that has the longest duration of any mammalian group in mammalian history. They're no longer with us today, but they're extremely interesting. They arose back in the Jurassic, under the feet, literally, of the dinosaurs, and they proliferated during the Mesozoic ecosystems, and they lasted well into the Cenozoic. And they've been a kind of classic, interesting topic in mammalian evolution as to what their lives were like as living animals. And so much of what he did for his dissertation work was to use various methods, field methods, laboratory methods, what we're going to hear about today, bone histology methods, to try to reconstruct aspects of these animals as living species, and particularly their life histories. And for his work, he got to go to some of the really interesting far-flung remote corners of Western North America to do interesting field work. We'll hear a little bit about that today, I think. And that piqued his interest not only in the things he's been working on with this particular unusual group of mammals, but also with the beginning of his ideas about the work he's doing here for his postdoc. So he's come here this past fall on an NSF postdoctoral fellowship, and will be here for at least 2 years and perhaps longer, depending on what the fates bear. So, Luke, welcome and thank you.
Right. Okay. Thanks, Catherine. Let me just get my microphone situated in my pocket. Maybe. And wave your hands at me or something if this starts rustling. I know that they have a tendency to do that. Okay, so can everyone hear me okay? All right, so like Catherine said, I'm fairly new to University of Michigan, and so I don't know many of you, but hopefully we'll get to know each other a little bit more as my time progresses here. And because I'm new, I don't really have any cool, exciting results to share yet from my postdoctoral work, so I wanted to highlight today some of the major findings from my dissertation research at the University of Washington and also with the Burke Museum. So, make sure my thing's advancing. So broadly speaking, my research aims to understand what are the mechanisms that are underlying some of these major transformations that we see in the early evolution of mammals. And in particular, I'm really interested in the Cretaceous period, which is the last kind of bit of the Mesozoic or the age of dinosaurs, and then how mammalian communities changed across the Cretaceous and Paleogene boundary. So that's the extinction the boundary that's marked by the mass extinction of non-avian dinosaurs and many of these other kind of charismatic faunas that you might have seen in like Jurassic Park, for instance. And then understanding how mammals radiated and evolved after that major mass extinction event. So when you think of mammals, most of you, if not all of you, are probably thinking of these kind of charismatic groups that I'm showing you here. So mammals today are very ecologically and taxonomically diverse. They span the range from fully aquatic whales to fully flying bats, and just about everything in between. But these major groups that you see up on this tree here, crudely drawn, is just a really thin snapshot of the diversity of mammals that existed in the geologic past. And really, the vast majority of mammalian evolution occurred much farther back in geologic time, in the Mesozoic and also, as I mentioned, in this early Cenozoic period as well. And it's in this kind of early days of mammalian evolution that you have the origin of the major mammalian groups that we would recognize, and the origins of their ecologies and kind of behavioral strategies that really characterize mammals as we know them, such as the evolution of complex chewing and high-frequency hearing. Most of what we know about mammal evolution, especially very early mammal evolution, comes from studying the gross morphology of hard tissues. So their skeletons, their skulls, their teeth, and we can learn a lot by studying these aspects of these animals' anatomy. So for example, teeth and other bits of the skull and postcrania can give us insights into the taxonomic diversity of these groups. The teeth and the jaws can also tell us about their dietary diversity, so kind of in the spectrum from more plant-based, more meat-based diets. We can also learn how different chewing systems evolved in major mammal groups. And in fact, this has played a big role in kind of the origin and diversification of different mammalian lineages. And then, of course, from their looking at their postcranial skeletons, we can learn things about their locomotor diversity and how that might have changed through time. But one thing that I was really struck by is that because so much of what we know about early mammals relies on these hard tissues, we don't know much about what I've often called soft traits. So things that we might know based on soft tissues or from observing animals that are alive today. And in particular, one thing that I really became fascinated by when I was a graduate student at the University of Washington is where or when in geologic time and where phylogenetically, so in which mammalian groups, did different mammalian reproductive strategies evolve. And so to tackle this question, I worked with a number of different folks at the paleobiology group at the University of Washington. The project was really started with myself and Meg Whitney, who's now a postdoc at Harvard, when we were both grad students at UW, and an undergrad mentee of mine who's now a master's student at Indiana University. And then along the way, a slew of other folks, including my PhD advisor, Greg Wilson, and some other grad students and postdocs at UW contributed to this work. So, diving into it, if we look at the three major groups of mammals that are alive today, we have the monotremes, which are the egg-laying mammals, today represented by the platypus and echidna. You have the marsupials, which most people are familiar with being things like kangaroos, opossums, koalas, the pouched mammals, so to speak. But the vast majority of mammal evolution or mammalian diversity that we see today are within placental mammals, the placentalia. That's the group to which we belong, your dog and cat, horses, whales, bats. Most of mammalian diversity is wrapped up in placental mammals. And the way in which these three groups are most easily distinguished, perhaps even in your own head, is based on their different reproductive strategies. So for example, monotremes, as I mentioned before, lay eggs, whereas both marsupials and placentals give live birth. However, among these live birth-giving mammals, the marsupials and placentals, they exhibit very different reproductive strategies. So marsupials have prolonged postnatal growth, which means they have a very short gestation period and that their young are born very altricial. So they're, as you can kind of see in this image here, they're little more than fetuses, essentially, and they develop almost entirely outside of the womb, often within the pouch. And that's so because of that, the marsupial mothers invest most of their reproductive resources into lactation to nurse their offspring. And that's in stark contrast to what we see with placental mammals, which have very prolonged prenatal growth, so these long gestation periods, and a relatively brief lactation or post-natal growth period. So their young are born relatively precocial or fairly well developed compared to those of marsupials. And so placentals, in contrast to marsupials, devote more reproductive resources into gestation to nourish their offspring. And so this divergence in life history strategies, reproductive strategies, between marsupials and placentals has often been termed the marsupial-placental dichotomy. Now, a bunch of hypotheses have been put forward to try to explain what caused this dichotomy and what the kind of long-term evolutionary consequences of this dichotomy were. But one of the most popular interpretations is that the marsupial strategy is essentially evolutionarily intermediate, or is the primitive state for mammals that give live birth. Because although marsupials share the trait of giving live birth with placental mammals. They also, in many ways, resemble monotremes in the fact that their young are very altricial and that they have these very prolonged lactation periods. And placentals, by contrast, are the only group of mammals alive today that give birth to these much more precocial neonates that have a much abbreviated lactation period. And kind of spinning from this idea that marsupials retain kind of the more primitive strategy or this evolutionarily intermediate strategy, it's been hypothesized that the marsupial mode of reproduction placed an evolutionary constraint on many aspects of their biology, such as their metabolism, their behavioral complexity, and also their morphological diversity. And that the placental strategy basically facilitated various evolutionary increases in their biology, such as increased metabolisms, greater behavioral complexity, in particular the evolution of social behavior, and also greater morphological evolvability. And so the story goes in this conception of mammalian reproductive evolution that when the placental strategy evolved sometime in the late Jurassic to the middle Cretaceous, so within the age of dinosaurs, that this essentially represented a novel evolutionary breakthrough that after the extinction of the dinosaurs 66 million years ago facilitated the rise to dominance of placental mammals that we would recognize today. But one of the problems with this story is that it's a story told almost entirely from the perspective of modern mammals. And so here I'm showing you again this cladogram with the three major groups. Eutheria is just the larger group that includes placentals. Metatheria is just the larger group that includes marsupials. And these two lineages last shared a common ancestor over 165 million years ago, so going way back into the Jurassic. And furthermore, although we only have these three groups represented today in the modern, there are numerous major groups of mammals that were very important components of mammalian communities and ecosystems back in the Mesozoic and also into the early Cenozoic. So if we want to understand how different mammalian life history strategies and reproductive and developmental strategies evolved, we need to get some understanding of how to interpret reproduction and development from extinct mammal groups, so looking into the fossil record. And so that's kind of the perspective upon which this project was based. And in particular, the group of focus for this particular talk is a group called the Multi-Tuberculata, or Multi-Tuberculates. So Multi-Tuberculates have the unfortunate situation of just being kind of a mouthful to say, but it basically just means many bumps, and it's in reference to their bizarre cheek teeth with these kind of longitudinally arranged rows of cusps. And as Catherine mentioned earlier, their claim to fame is that they're the longest lived order of mammals. So if you take a mammalogy class or read mammalogy textbook, that's often about as much as said about Multi-Tuberculates. They first show up in the middle Jurassic fossil record and don't go extinct until the end of the Eocene. So that's an evolutionary span of over 130 million years. And for most of that evolutionary history, they were by far and away the most abundant and diverse group of mammals compared to any of their contemporaries. They were also very ecologically diverse. So they exhibited a variety of different locomotor strategies and also had a variety of different kind of dietary ecologies. And they're also one of the only groups of mammals other than marsupials or placentals that actually went on to diversify after the Cretaceous-Paleogene mass extinction. So taken together, in the kind of early mammal circles, they're often considered to be the most successful group of early mammals. They're also a really good study system to get at these questions that I brought up at the beginning about understanding the causes and consequences of the marsupial-placental dichotomy. For one, that's because of their phylogenetic position. So they're phylogenetically outside of Theria, but they're also more deeply nested within crown mammalia than are the monotremes. They also have been inferred to have given live birth based on the structure of their pelvis, and limb bones that are well suited to histological analyses, such that I'll show you in a little bit, compared to all other early mammal groups, are actually fairly abundant in the fossil record. So the specimens are there to ask these sorts of questions. So, bone histology. this is really the way that vertebrate paleontologists can get at some of these soft traits that I mentioned at the beginning. Because obviously we can't see, how early extinct mammals are reproducing or developing, but the structure or organization of bone tissues and also the way in which they're vascularized reveal important clues about the developmental histories of extinct vertebrates. For example, animals that grow very quickly deposit highly disorganized woven bone that has a bunch of vasculature, so very porous and had a lot of blood vessels running through it. And it's from this relationship that we've been able to draw conclusions about other extinct animals. So for example, by studying modern birds and crocodiles and understanding how their life history traits are reflected in their bone histology, that's how folks are able to say anything about how quickly T-Rex grew or how old it was and things like that. But similarly, robust studies in the modern have not been done for modern small-bodied mammals. So as it stands today, we don't have a good understanding of how different reproductive strategies, for instance, in small mammals, are reflected in their bone tissue microstructure. And so we're missing this link to be able to infer what the reproductive or developmental strategies were of these early mammalian groups, which by and large were very small. Most of them were less than a kilogram. And so that's really where this project started. We first went and assembled a large modern data set of extant small-bodied mammals of both placentals and marsupials, and the phylogenetic relationships of those groups are shown to the left here. And in particular, what we were sampling was their femora, so their thigh bones, and looking at histological cross-sections taken from the midshaft, so the middle of the femur there. And here's just to kind of show you what those cross-sections look like. This is an array of our different samples. And in total, we ended up sampling 35 different species, 35 of which were adults and 22 of which were subadults. So these little dots next to the name are those species for which we had both an adult and a subadult. And we use this modern data set to then compare to a fairly large sample of multi-tuberculates. So we assembled about 18 multi-tuberculate specimens from our femora, I should note, and took sections from the midshaft of those femoral elements as well. And all of those specimens came from the Hell Creek area of northeastern Montana. So this Hell Creek area is famous for being where the first T-Rex specimen was found, and really has become a hotbed for understanding biotic change across the KPG boundary. And that's because in that area, the Hell Creek and Fort Union Formation are both exposed and were deposited both immediately before and immediately after the Cretaceous-Paleogene boundary. So all of our multi-tuberculate fossils in this study come from either the very latest Cretaceous or the very earliest Paleogene. And in total, as I mentioned, we had 18 different specimens that we sampled. And within that sample, there were 5 different morphotypes, which are roughly equivalent to different genera. So about 5 different genera represented. And when we looked at these specimens, what we found is that the primary differences between them, and similarities for that matter, were in the distribution of 3 primary bone tissue types. So what I'm showing you here is basically a cartoon of a cross-section from the midshaft of the femur. And these different colors that you see are representing these different tissue types. So the purple color in the middle is what we refer to as disorganized bone. And that's mostly woven bone tissue that's highly vascularized. And then on the inside in gray is this endosteal organized bone. And so it's composed of, well, it's very well organized, as the name suggests. And it's because it's composed of what's called lamellar bone. So essentially it's almost like plywood, so just kind of stacks of thin lamellae of bone. And this bone type has very little, if any, vasculature. And a very similar bone type also occurs on the outer part of the cortex, which we called periosteal organized bone. So again, like the endosteal organized bone, is mostly this lamellar bone with very little vascularity, but again is on the kind of the outer periphery of the bone cortex. And so when we looked and compared between our marsupial, placental, and multi-tuberculate samples, what we found was really striking. Marsupials are composed, their bone cortices are composed almost entirely of this periosteal organized bone. And only in some of the largest individuals do we get little slivers of this disorganized bone in purple and the endosteal organized bone in gray. And that was in stark contrast to what we saw in our placental sample. So in the vast majority of our placentals, we see this endosteal organized bone grading into disorganized bone at the midcortex, and then is sharply separated from this outer layer of periosteal organized bone. And so it forms this kind of sandwich pattern with these organized layers on the inside and the outside, basically sandwiching this disorganized bone layer at the midcortex. And when we looked at our multi-tuberculates, we saw the exact same pattern. So you see this endosteal organized bone layer grading into disorganized bone at the midcortex, sharply separated from this periosteal organized bone in the outer cortex. So the same sandwich pattern that we saw in placentals. So then the question was, what is driving this difference in histological patterns between marsupials and then the placentals and the multi-tuberculates? And what we think is going on is that those histological differences are reflecting important aspects of the marsupial-placental dichotomy. So to kind of explore this and lay out our thought process a little bit, I first need to introduce what the different autogenetic origins were of those three different tissue types. So disorganized bone that has been color-coded in purple is formed perinatally, which means basically just in utero or in the kind of early stages of postnatal growth. And to kind of show you that, what I'm showing you here is a cross-section from the femur of a very young juvenile Colombian ground squirrel. And you can see that that bone cortex is very highly porous, so has a lot of vascularity, and the bone tissue is highly disorganized. So it's this kind of webbing of bone that makes up the entirety of the cortex. And the organized bone is deposited much later in ontogeny. So what I'm showing you here, this is a cross-section from an adult American mink. And basically, the way bone tissue forms is it has to form on top of some pre-existing surface. And so what appears to be happening is that you have this disorganized bone precursor from perinatal growth that can then essentially act as the foundation for growth of either endosteal organized bone, which is deposited inward towards the medullary cavity or this big opening in the middle where bone marrow would have been. And this endosteal organized bone acts to basically in the remodeling of the bone cortex during growth and strengthening of that bone tissue. And then periosteal organized bone is deposited in the opposite direction, so from inside out, or what's called appositional growth. And the main kind of result of periosteal bone deposition, or this appositional bone growth, is that you're increasing the diameter of the bone shaft through ontogeny. And it's really this last point that this periosteal organized bone is what essentially grows the diameter of the midshaft that comes into play with understanding this difference between placentals and marsupials. And that's because when we look at the young offspring of a marsupial versus a placental, just to kind of illustrate that here, these marsupials are very tiny and very poorly developed compared to that of a placental mammal. And so a marsupial has much more growing to do to get to its eventual adult body size than does a placental mammal, even if that ultimate adult body size is roughly the same. And so marsupials, their perinates must greatly increase the diameter of their femoral midshaft to reach that adult size. And so what that means histologically is that although, like placentals, they're depositing this kind of disorganized bone in utero and shortly after birth, because they have to greatly increase the diameter of their midshaft, most of the bone tissue they're depositing through ontogeny is this periosteal organized bone, so this bone that grows outward. And that's a laser pointer. And in contrast, placental mammals, because they're much larger at birth and there's a smaller gap between the perinate and adult body size, they're essentially forced to basically reinforce or strengthen that perinatal bone matrix, and they have much less kind of outward or appositional growth to do to kind of increase the diameter of their midshaft. And so what that looks like histologically is basically you have this perinatal bone matrix laid down, mostly this disorganized bone, that's then reinforced through endosteal deposition of this organized bone. And then whatever growing they have to do to reach that ultimate adult size then deposits that periosteal organized bone layer. And so essentially what we think is happening between these two different histological patterns is that the placental pattern is reflecting remodeling of this perinatal bone precursor, whereas the marsupial pattern is reflecting much greater appositional growth needed to reach adult body size. And so essentially what this translates to is that the placental pattern is reflecting that placentals have much longer prenatal growth, prolonged prenatal growth period, whereas the marsupials have this prolonged postnatal growth period. And to look at this in a kind of more quantitative way, what we did is we went and using high-resolution photographs and ImageJ, we went through and quantified the proportions of these different bone tissue types across both our multi-tuberculate, our marsupial, and our placental samples. And what we saw, for example here, is that when we regressed the amount of this periosteal organized bone, so this outward deposited organized bone, against weaning age, we found a very strong and significant positive relationship between those two variables, such that placentals have a short lactation period and small amounts of this periosteal organized bone, whereas marsupials have long lactation periods and a large amount of this periosteal organized bone. And you might see in this plot that there's these weird kind of placental outliers that are clustering among the marsupials. Well, what was really exciting is that those actually turn out to be bats and primates. And among placental mammals, bats and primates have by far the longest postnatal growth periods and much longer lactation periods compared to any other placental mammal of similar size. So they are essentially more marsupial-like in their duration of postnatal growth. And sure enough, histologically and in terms, or histologically, they plot where we would expect them to be among the marsupials. So what explains this difference, histological differences between marsupials and placentals? Well, it seems to be reflecting in marsupials the fact that they have an abbreviated gestation and prolonged lactation period, and placentals seems to be reflecting the fact that they have prolonged gestation and abbreviated lactation. So then the next step is how can we take this to kind of infer the reproductive strategies of multi-tuberculates? Well, what we see is when we look at the proportion of these different tissues that we quantified to make that previous plot before, multi-tuberculates have a very similar kind of distribution of these different tissue types that very closely resembles that of placentals, and it's quite different than that, as you can see, of the marsupials here. So these are just stacked bar charts showing you the amount of periosteal, disorganized, and endosteal bone in the different specimens. And when we did ANOVAs to see whether this similarity or difference is significant, what we found is that placentals and multi-tuberculates are statistically indistinguishable in terms of their proportions of periosteal organized bone or disorganized bones in the cortices of the femora. And that both placentals and multi-tuberculates are significantly different from those periosteal and disorganized bone proportions that are seen in marsupials. And furthermore, because we have this relationship in the modern between weaning age and the amount of periosteal organized bone in the cortex, we could then take those periosteal bone measurements from multi-tuberculates and plug them into this regression as a way to estimate what their potential weaning ages might have been. And what we found is that the weaning ages that were predicted based on this regression model came out right around 30 days, plus or minus 10 days, which is fairly rough. But what was interesting is even with those large error bars, that put them squarely within what we see among modern small-bodied placental mammals, such as rodents in particular, and also some other groups such as shrews. And so all of this taken together, what we concluded is that based on this histological evidence at least, it seems that it's likely that multi-tuberculates had a placental-like reproductive strategy. And so this has major implications going back to our understanding of how different reproductive strategies evolved among mammals. For one, it suggests that although placentals have often been heralded as having this very unique and specialized reproductive strategy, that it's possible that this also showed up in this long extinct group of multi-tuberculates. And it also casts doubt on this interpretation that the marsupial strategy is evolutionarily intermediate. And instead, it lends support to a hypothesis that's been proposed by various authors that the placental reproductive strategy may in fact be ancestral for mammals that get live birth, and that instead the marsupials are the ones that have the derived or more specialized reproductive strategy compared to what early live birth giving mammals would have had. And if that's the case, then this intense maternal fetal contact that characterizes placental reproduction may have its origins tracing far back into the geologic past, perhaps as early as the early Jurassic or even further back in time. However, it's also possible that these similarities in reproductive strategies between placentals and multi-tuberculates arose independently. So it could be that marsupials indeed retain what is the more primitive reproductive strategy, and that both multi-tuberculates and placentals converged upon a similar mode of reproduction. And this wouldn't be entirely surprising given the fact that we already know that parallelism was extremely prevalent in early mammalian evolution. And some of the classic examples of this are in the evolution of tribosphenic-like molars. So they're basically the basic molar plan that you see in modern-day marsupials and placentals. Well, similar tribosphenic molars, or pseudo-tribosphenic molars, I suppose, have evolved at least 3 different times through mammalian evolution. And similarly, the detached middle ear, so the bones that form the middle ear in mammals that were once part of the jaw in reptiles, those have detached at least 3 different times through the course of mammalian evolution. So there are these very complex morphological structures that we already know have evolved independently in different mammal groups. So it's possible that a complex reproductive mode, such as that exhibited by placental mammals today, could have also evolved independently in both multi-tuberculates and placentals. But whichever of these interpretations is true, that the placental style of reproduction is ancestral for live birth-giving mammals, or that it evolved convergently in both placentals and multi-tuberculates, what our results do is that they challenge this idea that the placental style of reproduction is exceptional relative to other mammalian reproductive strategies. And indeed, this idea that the placental strategy was this novel evolutionary breakthrough that facilitated the rise to dominance in the Cenozoic, so after the extinction of dinosaurs, has largely been based on patterns that we can see in the North American fossil record. So if we look at the fossil record of North America, what we see is that after the Cretaceous-Paleogene boundary, which is marked by this red line here, we see that placentals diversify very rapidly in terms of both the taxonomic diversity, abundance, ecological diversity, whereas marsupial diversity plummets. Basically, only one species kind of limps through the KPG boundary. And so this kind of differential success across the KPG boundary has been attributed to the advanced reproductive modes of placentals relative to marsupials. But what this story neglects is the third important contemporaneous mammal group at this time in North America, the multi-tuberculates. And what we see is that throughout the Cretaceous, multi-tuberculates were more diverse and more abundant than both marsupials and placentals. And even after the KPG boundary, they rivaled placentals in terms of both taxonomic diversity and definitely in terms of abundance. And if the placental reproductive strategy had these evolutionary consequences for increasing metabolic rates, behavioral complexity, morphological evolvability, then perhaps the same sort of consequences were also in play for multi-tuberculates. And in particular, one thing that's been championed under this kind of school of thought is that the placental style of reproduction enabled the evolution of sociality and social behavior among placental mammals. Because when we look at modern mammals today, the vast majority of social species that we see are among placentals, and sociality is very rare in other mammal groups. And in fact, as a little anecdote, the earliest evidence of social behavior that has been found so far in mammals is actually in a species of multi-tuberculate from about 76 million years ago in Montana that seem to be kind of forming these colonial burrowing or nesting groups at a dinosaur nesting site. But back to this story. So it seems that multi-tuberculates may have had reproductive strategies similar to placentals, and that challenges that as being an explanation for why placentals went on to diversify. Because although placentals, take off and they go on to characterize the rest of the Cenozoic, multi-tuberculate diversity and abundance declines very rapidly about 55 or so million years ago. And what drove the decline and eventual extinction of multi-tuberculates is a hotly debated topic, and one that we can chat about if you all would like. But regardless of what drove this eventual decline and extinction of multi-tuberculates, what our findings seem to indicate is that it's unlikely that this differential success of placentals was driven by their unique reproductive and developmental innovations, or at least not those innovations alone. And stepping back a little more broadly, what I think is one of the most important implications of this is that we now have a mechanism of understanding, or at least approaching an understanding, of what the reproductive and developmental strategies of extinct mammalian lineages might have been. And so this represents kind of a new frontier of understanding early mammalian evolution, and it will ultimately help kind of resolve how different life history strategies evolved throughout the course of mammalian evolution. And with that, I've got plenty of time for questions, and I just want to thank a ton of different people, and in particular want to acknowledge that the multi-tuberculate specimens that I showed you were collected from the lands that are traditional territory of the Fort Belknap Assiniboine and Gros Ventre tribes, and also the Fort Peck Assiniboine and Sioux tribes. There's tons of different museums that contributed specimens to this project, the field crews who collected those specimens in the first place, folks for CT scanning, casting and molding, prepping slides, and many others for discussions. And I'll leave that up for questions. And I don't know if should I moderate the Zoom chat?
Hello?
Hello?
Oh, you can hear me, okay?
Yep, I can hear you.
So I have a quick question. So there's a possibility that marsupials strategy was derived, and if that's the case, what do you think could be the driver of that shift? What caused the marsupial to evolve their strategy, if that's derived?
Yeah, that's a great question. So there's been a lot of kind of speculation on this, and the one hypothesis that I think is the most interesting, or at least the one that's gotten the most kind of traction in the literature, is all relating to the fact that pregnancy is very costly for mammals in general, and placentals seem to have dealt with this by getting very good at having prolonged lactation or prolonged gestation. Marsupials seem to have dealt with this by just abbreviating gestation as much as possible. And one of the hypotheses for why the marsupial strategy evolved, if there was kind of a longer gestating ancestor.
Sorry, I can't hear you right now.
Oh, can you hear me? Can you hear me now?
The screen is also.
Oh, did I get muted?
Hello?
Can you hear me?
Oh no.
Oh. I think it's. Can other people hear me?
Now it's normal.
Okay, I'll hold it. Maybe it was just too far from my mouth. So anyway, to cut to the chase, the idea that's been put forward is that because pregnancy is so costly, and for mammals to have to undergo prolonged gestation, once they are pregnant, they're basically stuck with that developing organism in them until they give birth. And so all of the nutrients that developing fetus requires must be provided by the mother. And the hypothesis for why the marsupial strategy evolved is that by essentially abbreviating this gestation period and having the young developing within a pouch, they could much more easily kind of terminate the care of that young if environmental conditions got very tough. So they could essentially say there was a major drought or some serious limitation on resources. The mother could conserve her energy by getting rid of her offspring, not having to nurse them, and then try again when the next reproductive cycle comes around. So that's been one of the most prevalent ideas for why or how the marsupial strategy might have evolved derived from kind of longer gestating ancestor.
Thank you.
Yeah, thanks. Sorry about the tech confusions.
Actually, along those lines, since we have altricial and precocial birds, are there analogies in their environmental conditions and their reproductive strategies and life history strategies that inform this interpretation of the marsupials, of what drove the marsupials?
Yeah, that's a good question. So for birds, and I'm not a bird expert, but my understanding is that the altricial precocial spectrum on birds is much more similar to the altricial precocial spectrum for placental mammals. And so you don't have the extremely highly altricial neonates in birds that you do in marsupials. But what drives kind of the altricial precocial spectrum among modern mammals has been speculated about quite a bit. And one of the hypotheses is about rate of mortality. So, and this kind of falls into the R versus K selection strategy, but the idea being that where mortality rates are much higher, that giving birth to a precocial offspring that can go and get going essentially as soon as possible after birth is beneficial in that sort of environment. As far as what that might tell us about the marsupial and placental dichotomy, I'm not sure, just because I don't think it's exactly analogous even among fairly altricial birds. Yeah.
I'm curious just what made you want to study evolutionary history and mammals in particular?
So I didn't have any interest in any of this really until I took a human evolution class when I was a sophomore in undergrad at Colorado State, and got fascinated about there's this whole evolutionary past that I didn't know about for humans, and then I took a field class where I went out into Wyoming and collected early primate fossils and other early mammals from about 55 million years ago, and just became fascinated with early mammals and what were kind of the early precursors to mammal groups that we see today. And so it was really through that field experience and going out and collecting little mammal jaws in the Bighorn Basin of Wyoming that really kind of spurred my excitement and interest, and then it's just kind of gone from there. But yeah. Yeah, so I realized I should have been repeating the questions for the folks on Zoom, but the question was, how does the bone histology of monotremes compare to that of marsupials? Because monotremes, like marsupials, also have prolonged lactation and give birth to these highly altricial, these poorly developed neonates. So that's a great question. We weren't able to include monotremes in our study as much as we would have liked, mostly because they're fairly rare. In museum specimens, folks were not eager to let us kind of cut them up. But in comparing with the other ones that existed, we were able to get our hands on some of those photographs. And to our dismay, what we saw is that at least in some specimens of duckbill platypus, that they have this sandwich pattern that we saw in placentals. But the difference is that the bone cortex as a whole, so the specimens I was showing you earlier kind of looked like a big donut. There was a big gap where the medullary cavity was. Platypuses, their entire cortex is almost all bone. So they have just a very tiny medullary cavity in the middle. And on the one study that's explored what causes this pattern in monotremes, what they hypothesize is that because they're semi-aquatic, so living in the water, that they deposit all of this endosteal organized bone as a way to basically densen up their bones for buoyancy in the water. And that otherwise it would have been a mostly periosteal organized bone cortex. That said, when you look at echidnas, they don't look like anything, so echidnas are the other group of monotremes. They don't look like anything that we've sampled. Their bone cortex is very like woven and kind of looks like spider webby almost, and really looks like a juvenile placental mammal is what it looks like. And so of the two groups we see vastly different patterns that don't, at least at first glance, belie any sort of relationship to what their reproductive strategies were. So it's a great question, but I think right now it's just kind of a question mark until folks can actually see when in ontogeny these different bone tissues are deposited and whether it fits kind of our model for placental versus marsupial bone growth, essentially. Yeah, that's a great question. Yeah.
Going off that question, what's the fossil record like for monotremes and would you be able to look at the bone histology of extinct monotremes?
Yeah, so the question is, what's the fossil record for monotremes, and would you be able to look at the bone histology of extinct monotremes? So the short answer is it's very poor. Their true monotremes are perhaps known as far back as the Cretaceous, but that's fairly skeptical. And pretty much all we have of monotreme fossils until about maybe 20 million years ago or so, maybe even younger, are just a few isolated teeth and a couple jaws, so no skeletal elements. And even of those early monotremes, that fossils that we do have, they are mostly skulls and jaws. And you don't get more complete specimens until basically it's a platypus and echidna. So those kind of evolutionary precursors to what are today echidnas and platypuses, we don't have that fossil record yet. But there are a bunch of kind of groups in between monotremes and multi-tuberculates, or even between multi-tuberculates and therians, kind of looking phylogenetically, that do have full skeletons. And so that's one of our kind of next hopes, is to look at some of those other groups and see whether they lend support to the idea that this evolved convergently or whether it was ancestral. So monotremes probably aren't a good bet, but there are other early mammal groups that could kind of shed light on that. Are there any other questions on Zoom? All right.
Well, let's thank Luke then for giving us some insights into all the interesting, lively animals of the past. Thanks.
All right, thanks everybody.